Hello, everyone. We find out that our seminars are very helpful for our students as well as for other professionals in the field of functional food science to learn more about functional foods and bioactive compounds to prevent and manage chronic and viral diseases. Today, our presenter is Oladayo Apalowo from Mississippi State University. So he's a PhD student. And as I remember, Oladayo was among the students who was taking a course of Introduction to Functional Food Science in his university. I don't remember if Professor Ping or Professor Yang teaching, so maybe Oladayo can tell us. So his presentation topic is role of omega-3 fatty acids in healthy aging, metabolism, insulin resistance, and cancer.
My name is Oladayo Apalowo from Mississippi State University. I hope everybody can hear me.
Yes, we can. Everything is perfect.
So, yes, when I joined the Department of Food Science, Nutrition, and Health Promotion last year, I was introduced to functional foods by Dr. Ian Campbell, as Dr. Martirosyan has said. So, and I realized that there are great information that lies in the role of functional food to healthy aging, such as the management of chronic diseases, cardiovascular diseases, and cancers, and even viral diseases. And there pertains a wonderful, a potential effectiveness in the use of functional foods to precision medicine. So as you will be hearing in my presentation on the role of omega-3 fatty acids in healthy aging, I hope that this presentation will greatly benefit you. And by the time we are done, it's going to be a great deal to have spoken, to have interacted with you. Okay, so what are fatty acids? They are hydrocarbons that consist of the carboxyl group, and as well as many methyl groups. So in the slide, in the image on my right hand side, we can see this particular point. Let me share the screen. Okay, this slide. So we can see that this particular area that is denoted with red, and this particular functional group represents the carboxyl group, while the methyl end, starting from here, represents the omega group, and is named from the omega end, and it represents the omega group. So fatty acids usually have some kind of nomenclature, and one that has been used to classify them is their degree of saturation. When they have just one unsaturation, when they have one double bond in their chain, we call them unsaturated fatty acid. And when they have more than one, we refer to them as polyunsaturated fatty acid. But when there is no saturation, when there's no double bond in the chain, we just leave them as saturated fatty acid. So we also classify fatty acids in terms of their essentiality and non-essentiality. As we know in basic biochemistry or biology, that essential nutrients are those ones that cannot be obtained from the body, that cannot be synthesized, but they are obtained from diet, from the food we eat, and they are highly needed for the well-functioning of the body. Now, here we have just like a chart to represent omega-3 and omega-6. Omega-3, the examples are alpha-linolenic acid, which consists of three double bond, and so we refer to this as polyunsaturated fatty acid. From the omega end, as we can see here, if we start counting the chain from here, making 1, 2, 3, free. So that's why we refer to this as omega-3, because the first unsaturation starts from the third carbon atom on the chain. If we compare it to the omega-6 fatty acid, we will realize that the first unsaturation starts from the sixth carbon, and that's why we refer to it as omega-6. And as simple as this particular orientation may look like, they affect a lot of biochemical properties as well as biological functions of these different kind of polyunsaturated fatty acids. As I'm going to explain later on in my slide, functions relating to pro-inflammation, anti-inflammation, as well as the expression of some certain nuclear factors, nuclear elements, nuclear protein receptors are affected by this kind of orientation within these compounds. Through the function of desaturase and elongase enzymes, we see that alpha-linolenic acid, which is an omega-3 fatty acid, yields eicosapentaenoic acid, a 20 carbon atom fatty acid, as well as docosahexaenoic acid, which are very, very important and are typical examples of omega-3 fatty acid. We can see that their unsaturation is really, really enormous. For instance, docosahexaenoic acid has about 6 degrees of unsaturation. However, linolenic acid, a typical example of omega-3, omega-6 fatty acids, as a downstream product of arachidonic acid. Arachidonic acid is a precursor to several, inflammatory mediators. For instance, we can see that in its breakdown, it uses prostaglandin H2, while also we have downstream products of leukotrienes, thromboxanes, and prostacyclins. Many of these are pro-inflammatory. Okay, what are the examples of omega-6 and omega-3 fatty acids? So omega-3 fatty acids, they are usually found in fishes, marine fishes. For instance, examples such as tuna fish, sardines, titus. Yes, they are common sources of omega-3. And from findings, we realize that they prevent blood clots, causes dilation of blood vessels to enhance blood flow, and they decrease cell division, enhance the immune system, as well as improves brain function. While omega-6 fatty acid here, we can see that usually they are found in corn oil, in some foods such as flaxseed oil and the rest of them. And they promote blood clots, causes constriction of blood vessels, and they increase pain. They increase cell division, and this particular cell division is implicated in cell proliferation because in as much as they're able to increase cell division, it is to favor a particular end result, and that's angiogenesis, the formation of new blood vessels. Also, they depresses immune system and also have a negative effect on brain function. So as I said earlier on, fish oil is the basic source of omega-3 and is found in seafoods generally, salmon, smelts, sardines. They are high sources of omega-3, and they've been found to, several studies have reported their beneficial role in insulin resistance, glucose intolerance, which are the hallmark of diabetes, as well as cardiovascular diseases and some other diseases such as coronary heart disease. And many food supplements, many pharmaceutical companies, they have been able to develop supplements of omega-3 fatty acid. In fact, I remember at the time, I saw a particular soft gel-like product, which is omega-3, and a lot of people have recommended it, and it's fine taking it just for healthy body functioning and things like that. So now I want to talk about the reports that have come to play, especially on the relationship between omega-3 and insulin resistance. As I earlier said, insulin resistance is one of the major hallmarks of diabetes, and it occurs when muscle fats, that is the adipose cells and liver cells, are unable to take up blood sugar again. What happens is this: when there is more than necessary blood sugar in the bloodstream, basically, whenever there is sugar, there's sugar in the bloodstream, the body releases the hormone insulin, which attaches to the insulin receptor of these particular tissues, for instance, the muscle cell and liver cells, the muscle fat and liver cells, to uptake the blood sugar. But at a particular point, when excess blood sugar is sensed in the bloodstream, insulin will be produced by the pancreas, but the cells won't be able to take up as much as it should. And at that point, it is referred to as insulin resistance, because the cells will be insensitive to insulin. It will not be able to pick up the excess glucose in the blood. And that is what leads to diabetes. It leads to obesity and other vascular diseases. Now, this insulin is markedly consequential to the process of lipid metabolism, mainly via reduction of triglyceridemia by enhancing triglyceride lipolysis and fatty acid catabolism, increased high-density lipoprotein levels, such as the high-density lipoprotein cholesterol, and also being able to regulate the bad cholesterol, such as the VLDL, the very low lipoprotein cholesterol, and the low-density lipoprotein cholesterol. But there's a particular caveat, there's a particular receptor here that also functions through this particular cascade of reaction, in which omega-3 has been found to enhance. And this particular protein receptor is the peroxisomal proliferator activated receptor. Is a nuclear receptor protein, which is able to carefully regulate energy production, lipid metabolism, and inflammation. And it has been found that omega-3 is able to act upon this particular receptor. Now, I'm going to talk about a particular case study from Sitori et al. 1997. It was reported that about 935 patients with hypertriglyceridemia with no or with or without glucose intolerance or diabetes were able to be administered 1 gram of omega-3 for 2 months, 3 times a day. And after 6 months, it was found that there is a clear decrease in plasma triglyceride compared to the placebo. I mean, those people that did not receive the omega-3, there is a clear-cut difference. However, there was no marked difference in fasting blood sugar level, insulinemia, and OGT, that is the oral glucose tolerance. What did this study mean? That means omega-3 was able to reduce triglyceride levels, whereas it did not contribute to another emergence of glucose intolerance. It did not worsen the condition. It did not lead to another end result, but rather is able to ameliorate the incidence of hypertriglyceridemia. Also, this is an interesting study that was carried out on 60 male Western rats. Now, it has been used in several rodent models that the epididymal fat, which serves as the model for studying adipose tissue in rodents, was extracted, and then they were in this particular experiment. It was found out that 30 Western rats that were fed with sucrose-rich diet to induce insulin resistance were treated with fish oil. And the result was confounding because several biochemical parameters were found to be regulated by fish oil. And we know that the major component of fish oil is omega-3 fatty acid. Some of the parameters that were analyzed include superoxidase dismutase, uncoupling protein 2, as well as some inflammatory biomarkers such as TNF alpha, interleukin 6, as well as interleukin 12. And other parameters include the PPAR I talked about the other time. Now, from the results of that particular study carried out on the Wister rats, we realized that there is a control and then the sucrose-rich diet to induce insulin resistance, this is the group, and then those that were treated with a fish oil. In this particular first stage of the experiment, which really focused on weight, we realized that the body weight was reduced compared to the sucrose-rich diet mice. Now, when we come down to some plasma metabolites, specifically those relating to fatty acid metabolism, we realized that the triglyceride level was also reduced compared to the untreated group, that is the insulin-resistant group. And if we compare the fish oil treated group with the control group, we realize that it's almost close. And even in the result, there was almost no significant difference. Whereas compared to the sucrose-rich diet group, there was marked decrease when it was compared. Now, coming down to the last stage here, we see that the T-bars represents a particular metabolite to investigate oxidative stress, myelinaldehyde, and it was the T-bars was investigated, and we realized that the fish oil treated group was different from the sucrose-rich diet group, that is the untreated group. That means the antioxidant, the oxidative stress was markedly reduced. Though not up to the control group, but it was markedly reduced. There was a significant difference between the two. What about the tumor necrosis factor? We realized that it was markedly reduced. So there was a significant difference between the two, that is the treated and the untreated group. So this particular study makes us to realize that there was a positive, a beneficial effect of omega-3 as a functional food product. There was a positive effect, beneficial effect, even also on the inflammatory mediator here, interleukin 6. We realized that compared to the control, the fish oil treated group was much more reduced than the control. Now, these are the major antioxidant enzymes that were investigated. We have the catalase, glutathione peroxidase, glutathione reductase, as well as superoxidase dismutase. And these particular 4 antioxidant enzymes showed positive effects of omega-3 fatty acid in fish oil. We realized that all of the treated groups here for all of the 4 enzymes was reduced. There was marked reduction, as we can see. There was marked reduction compared with the control. So the control and the treated group almost had no significant difference in their values, while the untreated group, that is the induced group with sucrose-rich diet, was statistically different from the treated group. Now we have here the result of a Western blot that was normalized with beta-actin. So the PPAR here, for the 1 here represents the control diet, 2 represents the sucrose-rich diet, and then the number 3 represents the treated group with fish oil. we can see that from this expression, it is clearly seen with the band here that it is highly expressed compared even to the untreated group. So that means that PPAR was increased in the group treated with fish oil. Now, finally, for this particular stage, we can see that omega-6 here compared to the control diet was markedly increased. Also, omega-3 too was markedly increased. Now, the ratio of omega-3 to omega-6 has been a threshold, a marker to discuss the potentiality of whether a particular person is taking the right amount of the right kind of fatty acid. So we see that the ratio here is increased. Had it been the ratio of omega-3 to omega-6 is reduced, then there is a problem, as we have seen in the sucrose-rich diet. But since it is increased, we can see that there is a beneficial effect of this particular fish oil in ameliorating and not allowing the condition caused by sucrose-rich diet to exacerbate in this Wister rat. Now, coming to cancer, we realize that cancer has now shifted from the previous definition of maybe a particular disease. No, it is now found out that cancer is a group of more than 100 diseases affecting the body. As we have found out in our introduction to functional food science course, that it is a slow and complex multi-step process characterized by acquisition of novel biological capabilities which regulate cancer progression via sustaining cell proliferation signals. Number 2, inhibiting growth in suppressors. Number 3, apoptosis, that is cell suicide. Number 4, promoting angiogenesis and inflammation. And lastly, activating cascades of proliferation known as metastasis. Now, what have we heard about omega-3 and cancer-related inflammation? The first thing is that eicosanoids from arachidonic acid, they are key mediators of chronic inflammation. However, eicosapentaenoic acid-derived eicosanoids, they are usually less inflammatory than the arachidonic acid-derived eicosanoids. And we realize that membrane phospholipids, they are usually competed for by omega-3 or omega-6 fatty acids. So it depends on the one that is able to have more levels of phospholipids. If we have, remember that phospholipid usually has a backbone of glycerol. The first carbon atom is esterified with saturated fatty acid, while the C2 is esterified with unsaturated fatty acid, which may either be omega-6 or omega-3. And then the last carbon is esterified with a phosphate backbone. That's what makes up a phospholipid. So the C2 is the position at which we find whether this particular phospholipid we initiate a downstream product of inflammation, a downstream product of or a wrong byproduct. At that particular position too, it can either be esterified with omega-2, omega-3, or omega-6. And once it is omega-3, then all things is fine. But if it is omega-6, then there's a problem. That means there's going to be a higher expression of inflammation. Yeah. So we've been able to find out that some studies show that omega-3 fatty acid inhibited the synthesis of pro-inflammatory molecules, such as tumor necrosis factor, interleukin-6, monocyte chemotactic protein, and it was done through the activation of a particular protein receptor, GPR120, which is known as a sensor of omega-3 fatty acids. Now, this is a complex chemical pathway And we can see that all these particular metabolites, well, to like say metabolites, this particular, okay, yes, metabolites, all of them too, we found out that they are pro-inflammatory, while those in green, they are anti-inflammatory. That is, they are helping to prevent the process of inflammation. At the first point here, we see omega-3 inhibiting this particular step, while PPAR is inhibiting nuclear factor kappa B. We see GPR120 here, a sensor of omega-3 fatty acid, and we see two particular interesting downstream products of alpha-linolenic acid here, eicosapentaenoic acid, and docosahexaenoic acid. And we see a downstream product being inhibited, inflammation. So NF-kappa B has been found to increase inflammatory cytokines, chemokines, adhesion molecule, cell proliferation, apoptosis, morphogenesis, and differentiation of blood vessels. Now, if these particular processes are inhibited, then I think we may be able to bring cancer into check. So I have here a summarized chart, which I found in the Introduction to Functional Foods book on page 316, that gives the a general overview of the major pathways that omega-3 polyunsaturated fatty acid is able to affect. The first one is inflammation here, how it affects the expression of cyclooxygenase and lipooxygenases through the upregulation of GPR120 that I said earlier, PPAR, and the downregulation on NF-kB, and thereby decreasing the incidence of inflammation. Also, we realize that angiogenesis, which is usually a particular pathway of blood vessel formation through the upregulation or the production of the VEG, vascular endothelial growth factor. We realize that omega-3 is able to upregulate the production of resolvins and then lead to the downregulation of cyclooxygenase as well as PGA2 prostaglandin A2, thereby inhibiting the production of VEGF and thereby stopping or reducing angiogenesis. Angiogenesis is typically a particular method by which cancer cells are able to form blood vessels that will be able to supply nutrients to during cell division. You know, cancer cells, they highly proliferate. So as they are generating new cancer cells at distal sites in the body, they also need blood vessels that will help to reach to that site so as to allow coordination and crosstalk in the cells. Also, we have survivor. You know, cancer cell will not die. They won't want to die. So they don't usually undergo apoptosis. They always look for mechanism to prevent the occurrence of apoptosis. Because if cancer cells dies, then the person is cured of cancer. So omega-3 fatty acid is able to enhance the production of P53, hormone suppressor factor, and also reduce beta-catenin, and also reduce the mTOR, whereas increasing caspases and then increasing apoptosis, as well as autophagy, which are mechanisms by which programmed cell deaths take place. So as a summary, I have a lot of processes here in which omega-3 fatty acid is able to influence. It decreases very low density lipoprotein from liver. It lowers serum triglycerides, as I said earlier on, and also decrease arachidonic acid derived prostaglandins, and also metabolize to omega-3 anti-inflammatory mediators like resolvin and protectins, which are able to also help in anti-inflammation. So in summary, the effect of omega-3 fatty acid, either marine-based or plant-based, has been shown to exhibit no adverse effects on cardiometabolic incidences. Cancer development involves multiple genetic alterations in signaling molecules and the dysregulation of associated signaling cascades. Experimental and epidemiological studies have revealed a positive effect of omega-3 polyunsaturated fatty acids on cancer prevention and treatment. However, major mechanisms for the role in quenching adipose fats and reducing triglycerides levels still need further studies, especially to be able to determine and come to understand the direct mechanism of action by which functional foods is able to mitigate the effects of insulin resistance and glucose intolerance, and in this case, omega-3 fatty acids. Lastly, clinical trials investigating the direct influence, that is the targets of omega-3 on primary or advanced cancers are needed. Take, for instance, colorectal cancer, sigmoid, or transverse. These are all expressed in the abdomen, in the abdominal cavity. So does it mean that a particular drug like omega-3 will be able to affect in the same way, in the same dose, all these three kinds of cancers? That we don't know yet. And that is why there is a need for further studies. There is a need for more clinical trials. There is a need for more experimental trials to be able to understand the roles of omega-3 fatty acid, as well as some other functional foods that could be of that could be highly effective in regulating the effects or the incidences of cardiometabolic diseases as well as cancers. Thank you very much for your time and listening to my presentation. I would like to take some of your questions.
Thank you, Oladayo, for the great presentation. And let's see if anyone have questions. But I would like to ask you maybe a little bit out of this presentation. So do you know if there is any particular product, healthy food product or functional food product, which really can control that, which is recognized? For example, we know that there's a lots of products, over 1,000 products in Japan approved by FOSHU, and among them there is several which could be related to this to the topic that you presented. So how to control, for example, how to control glucose level. Yeah, if you have any comment on this.
Yes. There are so many functional food products that have been marketed and have been tried, and there are a lot of recommendations for them. In fact, during one of my presentations in spring with Dr. Ian Campbell, we investigated and talked about a particular product which I presented from Nigeria then, that was used, and a lot of people actually recommended it, and it was used for the management of diabetes, so it won't get out of control. Although the issue with some of the products is their mechanism of action not yet being understood, the exact pathway by which they act. And that is why there's still a lot of things to do as scientists to be able to come to understand the mechanism and the particular active ingredients that is working in these products. Take, for instance, omega-3 fatty acid that we are talking about. It is found in fish oil. But for instance, the mouse study that was carried out, the experimental study that was carried out, it made use of fish oil, not exactly omega-3. Yes, and there could be a lot of factors. We can see that it could be to be able to get the right amount of omega-3. That would take time, and then would getting that particular omega-3 out not prevent the biological effects that is going to take place in the body. That it's not even exactly omega-3 fatty acid that is carrying out these particular biochemical processes. It is talking about the eicosapentaenoic acids, the docosapentaenoic acid, and other downstream metabolites, which, when taken as whole food, it might be inside the system that this particular metabolites will be able to express and have their biological effects. So there are a lot of food products that are being used. We have other ones like flavonoids. Flavonoids, in fact, there are a lot of products that have been supplemented in foods. Flavonoids, anthocyanins, and the other kinds of them that have been found to be highly effective in ameliorating the incidence of diabetes, insulin resistance, glucose intolerance, and even high blood pressure, many of them.
Okay, very good. So at some point, you're saying that there is some good candidates, but in a particular case, so it wasn't a clear mechanism of action. So that's why we cannot claim that that's functional food, which is very right conclusion. Yeah, absolutely. And the good thing here is that as functional food center, as well as for sure in Japan, so recommending, it's mandatory, I would say, to find out mechanism of action of bioactive molecules which are representative, which are responsible for particular effect. So certainly, yes, we have to find out molecular mechanism. Otherwise, it's going to be difficult, especially when we speaking about food, where there's lots of different ingredients. So then this is not going to be correct without finding out exact bioactive compound and exact molecular mechanism, how could be working there. Yeah. And in addition to that, I would like to say, at least we know that there is, in FOSHU, there is fiber-cell 2. Fiber-cell 2 have lots of different application, but particularly that has also application for glucose control. And they find out that kind of 4 gram, 6 to 6 gram, particularly for glucose control. And then for different other options there for cancer, for other chronic diseases, different amount of fiber-cell 2. So they done excellent research. They come up with almost all steps they done. The only thing that we mentioned that that should be done in future. So that was epidemiological studies and aftermarket studies, because they're producing a product and then several years in market. So it is much better to see what's happening, because one thing when you have only clinical trial with 100, 120 people, but another thing when then now you selling product via these stores, and we have to do market and see how that was really effective in open society, but not in controlled clinical study. Okay, let's see. Here is one question. Thank you, Ebenezer. Have you researched fenugreek for improvement of metabolism and help for diabetes and metabolic syndrome management? So if you use fenugreek.
No, I haven't done anything about fenugreek yet.
Yeah. Yeah. How about I see that we had a person here from chia related company. Let me see if he's still here. Yes, Fernando from a world of chia. So you know that chia is providing the excellent, one of the best resource for omega-3.
Yes.
And really omega-3 is, I mean, chia is having so great amount of, and myself, I am also using chia as a resource of omega-3. You know, it's very easy to use with different recipes, and simply one teaspoon is going to be enough to do the job, you know. So if you know any particular might be product related to chia might be, so omega-3 from chia. My question was to you, if you find out might be some product,
okay,
a healthy product, I mean, it's difficult to say functional food product until we do not know that they really conformed that they have functional food, if they go through all steps that we mentioning, like how to create ideal functional food. But if you know any product in market which is coming from like chia, from plants resource.
Yeah, chia seeds, I've heard a lot about chia seeds from, there was a time my advisor and I discussed, and he just mentioned to me about chia seeds, that he's taking it and things like that. Though I have other sources of omega-3 that I take, which mainly they are fishes. I take a lot of fish, yeah, specifically sardines and Titus. So and yeah, and I think chia seeds, we have it in a lot of oils too, and then yeah. So,
yeah. So I would like to say that really, you know, chia is why it's good, because it is available now, at least in United States. So we can see that even in big stores like Costco, I was purchasing from Costco, and it is not expensive and it is organic. So, and it's easy to use. Simply you put in hot water. For example, I'm using to make my coffee. So simply adding.
I think I'm going to try it out.
Yeah, skipping a few minutes, like 5 minutes, 10 minutes, so they're gonna pop up, and very good. And in some restaurants or kind of cafeteria, so they also making kind of stuff, drinks with chia or smoothies. So yeah, that's for our participants and for you. Any other question?
I think someone is signifying. Someone is raising up hand.
Yes, thank you for the presentation, Ebenezer. So I have a question about like an optimal amount of like omega-3s that you'd recommend or that you've seen, because can you hear me, by the way?
Yes, you can.
Okay,
this
perfect.
is Kevin, right?
Yes.
Let me try to yeah, if you could start video, yeah.
Sure. So, you know, an American diet or just Western diet is pretty high in the like omega-6 to omega-3 ratio. So if people don't really change their diet too much, but you know, they take their supplements, right? Do you think that has like a real anti-inflammatory effect, or is from what you've seen, like people really have to change their diet in order to have those sort of positive effects come through?
Yes,
at least in humans, yeah.
Yes, I think one thing to come to understand about functional foods is the balance. The balance is key. Balance is key. So we can't completely rule out the place of omega-6 in our diet. It just needs to be in check. Just needs to be in check. So that's the trick about functional food. For instance, take selenium. selenium overproduction in the body can be detrimental, while its underproduction too can be very, very consequential. Some drugs, when you take them overdose, there will be problem, and you should take the right dosage too. So we need to come to understand why we are using it, what it's going to do, and our body status of any particular mineral we are going to take. Many of the foods we take, are they rich in omega-3? Are they rich in omega-6? What's your kind of diet? So these are things we need to first find out before we start taking a food supplement, so that we don't get overproduction of their downstream effect. We don't get too much into our body and then see the wrong effect. I don't know whether Dr. Martirosyan has...
Yeah, I think that's correct. And the question was very right question, because if you don't have a right diet, and then we simply using one bioactive compound, omega-3, and how we using that, again, this is also questionable. So we have to think about changing ratio, because that ratio really changed much. So that ideal ratio, omega-3 to omega-6, should be 1 to 2. 1 to 2. So 1 portion of omega-3 and 2 portion of omega-6. But nowadays, in many countries, even developing countries, that ratio shift up to 1 to 16, and so more than 1 to 10. So then this is a real good question. So having same chia as a good resource for omega-3 daily, so this is going to change. And certainly having a fish, this is very good. And why not? Yes, fish for DHA, DHL, and also EPA, and yes, chia for ALA. So then we have a whole combination of omega-3. So in that, as it coming from food, it's going to be much better than simple supplementation. And also to let everyone know that we simply considering functional food when it is coming as a food, but not like pill or supplement. And that's the thing. There's a lot of investigations, and where we saying that this vitamins, that vitamin mineral is very helpful, and there's a research, but as it coming to real life, so then especially much easy to do supplementation, so then it is not working. Actually, yesterday I was having a very good meeting with the president of IFT, and that was one of the topic. And they also like so much about functional food, and asking our company organizing might be some seminar in next IFT conference. So this is really very important to understand that we have to get mostly as a real food. So yes, when we investigating, yes, we have to investigate molecular mechanism, bioactive compound, isolate, to do good research, figuring out that exactly this bioactive compound is responsible. But at the end, we have to come up with real food. So even if we have a powder, so then we have to be able, so adding water and making a soup or whatever. And even more, in European country, it is standard a rule that yes, even if there is approved that this bioactive compound is very useful for kind of thing, but then if you making a food new food product, you have to prove that really everything what you done before now is applied to this particular food. So that's another step when we saying creating a food vehicle, choosing right food vehicle, and we have to prove that that bioactive compound within new product, it works. And how that works? Because it could be in food vehicle, could be many other ingredients, how they're incorporating, etc. I think we answered on that question. So here is Fernando recommending us good article about chia seed. So I don't know if everyone can see that. If not, so we can send this to everyone. So more likely everyone can see this. Yeah, that's it. Oladayo, thanks so much. If you have any other comment, some recommendation, if yes, you are free to do. If not, so we're gonna finish our today's seminar.
Yes, thank you everybody for attending and listening to the presentation. And I encourage you also to participate in some of the courses outlined in the Functional Food Center website. It will really be helpful. Some of the books too, they are good.
Yeah, look like we lose connection. So until it's gonna be, his internet gonna work, I would like to say, yes, this is one part for how to become functional food scientist. So this is one step, and one other step gonna be for Oladayo and other people.
Not my connection. Sorry for that. I don't know what happened.
It's okay. It's okay. You can continue.
Okay, so I was talking about some of the books available on the Functional Food Center website that can be helpful to us. So if you can stop by and just get some of the books, check them out, and some of the courses too that you can take that can better your understanding on functional foods. Thank you very much, everybody.
Yeah, I was, as it was muted, so I was explaining how this certification works, that this is presentation is one step, and one other step gonna be we share it with Oladayo, very important document with about 25 pages, how to come up with ideal functional food product, what has the definition of functional food, what are the regulations in Japan and in other countries. So then one more meeting I am going to have personally, or some other functional food certified scientist. So we're simply going to discuss with him how he understanding those important stuff, and then he's going to get his certification. So listeners also know about how this works. Okay, so then thanks so much, Oladayo, and thank you to all participants, and have a very nice day.
Thank you. Bye-bye.